So we're really delighted to have Liberty Hamilton back for a second year. Some of you actually may have been here in Seattle last year, so might recognize Liberty. Liberty is actually one of my colleagues here at UT Austin in Departments of Neurology and what is communication disorders? I always get that wrong.
Yes, and now they actually just changed their name to Speech, Language, and Hearing Sciences. So
to make it even more confusing, just to.
Yes, exactly. Yeah.
Cool. Well, so Liberty's going to tell you about some of the work she does and about just playing around, I think, with ECOG and EEG data in Python. So we're delighted to have you, and take it away.
All right. All right. Thank you so much, Tal. As Tal said, I am also a faculty member at UT Austin, and study speech perception and production in naturalistic environments. So I'm going to be sharing some work from the Jupyter Hub. So I will share my screen on that shortly. I'm also going to share a link to a HackMD notepad that I would like folks to use in order to be able to have some interaction with you a little bit. So let me share my screen here. So, all right, so I will be showing you, hopefully here you can see the screen that is the HackMD website. I also have Jupyter Lab open over here. This is the Neuro Academy Hub. Tal, let me know if either of those look like they're both showing up. Hopefully both of those tabs are showing. Okay, so if we go to this first one.
Yes, sorry, they're there. I was muted.
Great. Okay, perfect. So what we'll be talking about today is using a tool called MNE-Python. And this is a very nice open source software package written in Python that is used for, in their words, exploring and analyzing human neurophysiological data. So that includes EEG, MEG, and IEEG in a more proprietary way, I would say. IEEG stands for cranial EEG, which is the invasive component of this invasive and non-invasive EEG data that we'll be looking at today. And this package, again, is developed by a lot of contributors all over the world. And I will mostly be using MNE-Python, but some of this tutorial will also cover some plots that you can make in Matplotlib. And of course, we'll be using NumPy to do some array manipulation as well. So if you've been to any of the other tutorials on Matplotlib or Python basics, that should also be helpful for this tutorial. And I'll kind of give you an idea of what sorts of things you're able to do with MNE. And then if you want to branch out to something more complicated or just a little bit different, then you can by basically exporting the data from MNE and looking at it in something like Matplotlib. Okay, so hopefully some of you have started looking at this. So I would love it if you are part of this, this Zoom webinar, if you could write in the HackMD sort of scratch pad here. You can write your name, sort of what your status is. Are you a PhD student? Are you an undergrad? Are you a postdoc? All of that sort of stuff. I just want to see kind of who is here and what sorts of methods people are using in their day-to-day lives. And so some of the plotting here, depending on your background, may be more basic. But if you have never MNE-Python before, I will kind of show you some of the things that MNE-Python can do in order to look at things like evoked potentials, to look at stimulus-locked data, and all that sorts of stuff. So it looks like we have a good crowd here so far. Lots of EEG folks, a couple ECOG, EEG folks, MEG, fMRI. I know there's a big fMRI contingent here at Neuro Academy, but also trying to branch out so that we talk about all sorts of different methods. And here, actually, as I said before, MNE-Python is, I believe it was originally developed for MEG and then EEG. And now there have been more and more contributors who are working with intracranial data, which has its own set of difficulties because a lot of it is not particularly standardized. And so if you went to any of BID's tutorials, you'll see there have been efforts to try and standardize things a little to try and avoid this. But all right, so I'll just give a little bit of time. And all right, great. And the last part was, what made you interested in this tutorial? Maybe just sounded really cool. Maybe it looks like there are people who are interested in transitioning from MATLAB to Python. And hopefully we'll show you some of the things that you can do with this Python tool that if you have used things like EEG Lab, for example, or some of the vision analyzer software, you'll see that some of it is similar. I'm not planning on talking about pre-processing in particular today, but of course, M&E is a fully featured software product. So you can go all the way from your raw EEG data or intracranial EEG or EEG data all the way to artifact rejection, independent component analysis, filtering, plotting analysis, all of that, all in one thing. All right, so looks like we are great. Okay, so it looks like we have a good group here with lots of sort of diverse backgrounds here. So that's great. So you can keep filling that in, and I'm going to switch over to this Jupyter Hub. So this is the NeuroHackademy Hub. Hopefully you have already seen how to use this in some of the tutorials. If not, when you first launch it, and you may have to, if you find that some things are not working well, you may need to go into the Hub control panel and restart Hub, hopefully not. But my tutorial is in curriculum, we visualizing EEG, and I know it's Wednesday, but I like that it sounds like this is a, this is a thing we're doing together. We're visualizing EEG. And you will just open EEG, the Python Jupyter notebook. Okay, so, all right, so I'm also going to increase this font just a little bit because I know this can be hard to see for folks. So what we are going to do in this tutorial, we are going to load some sample EEG data. We're also going to some sample intracranial EEG data. These data sets are both from my lab. They're actually, as of now, unpublished data. So you get to play around a little bit with some of the things that we're working with right now. Unfortunately, there isn't a ton of intracranial EEG data out for download, especially if you're looking for things like sensory neuroscience data. So I have seen quite a bit of data sets that are mostly related. So if you're looking to plot propagation of seizure activity, you can find that a little bit more easy or a little bit easier than task related intracranial EEG. In any case, I'm providing this. That should also be in data directory if you go up all the way, but this notebook will load it automatically. All right, so you'll be able to load the data, which is from our lab. We're going to plot the raw data and see what that looks like. Some of the features, I will say, in the Jupyter notebook are not going to be as full featured as if you were using an interactive IPython session, for example. So the raw plot is going to work, but if you were actually trying to scroll through the data, you would probably want to do this in an interactive book setting or with just the Python terminal. We'll be plotting of the data, which is something I often do to check data quality and to check what filtering may or may not have happened on the data. And then we'll start doing stuff where we're actually looking at how to divide our data up in epochs. So if you have some kind of task, then how do you extract the task-related information and segment your data so that you can have the EEG or the ECOG that occurred at a particular time frame. And you can average a stimuli with specific characteristics, or you can average all of them. You could do lots of things. And then after that, yeah, we'll go into plotting the evoked activity in response to these different task conditions, both by ECOG channel, averaging across channels. I'll show you some of the ways that we're able to plot EEG activity on the scalp as a topo map, and ways of customizing plots. All right. Of course, there are also other resources from the MNE Python website. There are many tutorials that cover some of this same information. Sorry, this is not needed. Some of this information mostly separately. So, you know, feel free to look at those if you feel like you want more, but this should at least give you a taste of how this works. Okay, so, all right, so for the next part, what we're going to do is hopefully if you're following along in your notebook, one thing I would check is that you have this line at the top, pip install matplotlib equals 3.2. This is because for unknown reasons, the current version of matplotlib is being a little funky with MNE Python for just one of the plots. So if you have the version of the notebook that does not have that line, just go ahead and add that to yours. Okay, so what we're doing here is we're just going to load in some data, and basically this whole tutorial is kind of going to be me live coding here in the notebook, talking about different ways that can change the plots or sort of redo them in some interesting way. But you can also feel free to ask questions through the Q&A, or I'll try and also monitor the HackMD pad over here. Okay.
And I'll keep an eye out on the Slack channel in case anyone asks.
Oh, wonderful. Okay, thank you. All right, so here, as I said, we're to be importing just a few libraries, mostly MNE, NumPy, and Matplotlib. These are the data sets that we're working with, and I've just placed data sets in a dictionary, and you don't have to do this. This is more just because I have these two data sets that are actually people to the same sentences, but it's different modalities. So one is with scalp EEG and one is with intracranial EEG. And so for this first set of things in the dictionary, these two are the pre-processed files, the EEG file and the ECOG file. And I'll show you some of the characteristics of those. In this, I decided to work with pre-processed data. So that was just in an effort to simplify things a little bit, because pre-processing, I could also give an entire 90-minute talk on. There's many different aspects to rejection and filtering and lots of things to worry about there. So this is pre-processed. These two items are the event files, and we'll talk about those in just a bit, but they basically say the timings of when our stimuli occurred. And then we have this list, which is going to list the names of the stimuli that we are using. All right, so I'll go ahead and run this. And it says, okay, we're uninstalling Matplotlib 3.3, successfully installing 3.2. You'll probably see something like this as well if you've restarted your hub. And if you end up having any problems with the other tutorials because of Matplotlib, you can just go ahead and restart, and then this installation affect anything else. Okay, the thing that we're going to do is we're going to get some information from these stimulus files and using a function called gen from text, which basically takes our stimulus file. It's a comma separated file, so that's what this delimiter means. and we're going to take the 0th column and the first column, and we're going to load those in, and we're going to create a dictionary of event IDs and corresponding numbers. So I'm going to add one last here to just show you what these are like. So printed out event names, and we print out event numbers, and we run this. Okay, so this first one is a list of all of the stimuli that we used in our experiment. And so these are named accordingly. They all have these kind of alphanumeric strings and SI, and then another set of numbers. .wav is because they are audio files. Scroll down, see that we have lots of these, so lots of stimuli that have been played. And then these are just the corresponding event IDs, and that will become important later when we are looking at segmenting the data according to these specific sentence types. So each one of these WAV files is one sentence. All right. So here, what I'm going to do is I'm first going to go through this tutorial with the EEG data set. And again, like I said, this is looking at some data sets that are from my lab, all collected at UT Austin or in collaboration with clinicians here in town. And these were collected from 2 different participants who were listening to sentences from the Timmett acoustic phonetic corpus. So for those of you, if any of you work in speech adjacent research, you might have heard of this. Otherwise, I understand if not. This actually stands for Texas Instruments Massachusetts Institute of Technology. And this was created as a database of spoken sentences. And it has a number of talkers from throughout the United States, both male and female talkers. And it's been used to train speech recognition algorithms. It's been used in linguistics research, neuroscience research, and more. And this is kind of a nice data set because each sentence is relatively short, but it also gives you a good distribution of different types of accents, different types of voice quality. So the pitch of the different people's voices is really quite varied. But you can look at things like pitch selectivity, or you can look for selectivity in spite of what the pitch is. So the idea here was to get a sort of distribution of all of the phonemes that are present in English, including some of the ones like je, for example, in Azure, or collusion is my other favorite one lately. And those normally occur in a very naturalistic stimulus that often, but this tries to sample those a little bit more often. Okay, so as I said before, the list of these stimuli is in stimuluslist.cs, which we can actually also look at here. The data are in data, EEGViz, and so we've got event numbers, event names for each of these. And one thing I'll add that I didn't say before, that each one of these stimuli starts with either an F or an M, and that corresponds to whether it was a female talker or a male talker. The rest of the code also has a meaning having to do with other characteristics of where the speaker is from. If you're interested in that, you can read more. Timmett website I have also linked. You can read about that database. So we're going to use that later to show different ways of splitting up the data. Right, so that's stimulus list. The other thing that you would need is the stimulus timings. And this is something that you may be getting from your hardware. So some types of hardware recording amplifiers will have an additional trigger port, or they will have some way of locking individual sort of pulses for when each of your trials occur. We actually use a slightly different way of finding the timings of stimuli, recording the audio itself into the EEG system, and then using what's called a match algorithm to find where each of those sentences start and stop. And we use those timings. And I'll do in the EEG file, if we look at that, this is a tab, and this has basically the start sample, the stop sample, and an integer ID that corresponds to which stimulus was heard by this person from this sample to this sample. And so you can see we've got a lot of trials here over many different samples. And so we could, for example, okay, this first trial was stimulus 105, and we can see that stimulus 105 was a female talker, this FTAJ01320. Okay, so that's not super important, but I did want to show that just in case you are working with evoked data and are thinking about stimulus timings, kind of the format that you would need your event file to be in. And notice that it's not in seconds, it's not in time, it's in time. So this should be based on whatever the sampling rate of your data is. So if you're recording EEG at a 128 hertz sampling rate, then this thing divided by 128 would be the seconds of the start time for the first stimulus. Okay, all right, so just a little bit more about the EEG and ECOG data before we get started. In my lab, we use the Brain Vision ActiChamp system, and we have a 64-channel high-density EEG setup. And as I said before, these data are part of an ongoing project in our lab. So thus far on, we do plan on releasing these with the paper eventually. And I did just want to mention that you can find similar larger data sets from another speech study by Broderick et al., for example. This is from Er's group. Or actually, Brad Wojtek's lab had a list of open physiology data sets. So that's quite nice. And MNE Python does have readers for quite a few of the different systems that are used for recording EEG. So hopefully there's a way to get your data in there. For ECOG, it's a little bit different. So our ECOG data was recorded from 106 electrodes, and this was across multiple regions of the brain while our participant was listening to commit sentences. And this is a sort of smaller data set than the EEG data, so faster to load. And we recorded a number of different areas that are labeled in the file according to acronyms. So you'll see these, and if you're kind of thinking, what the heck is ROF? Then you can look at this table, and the general location of those electrodes would be in the right orbitofrontal cortex. Just for fun, I guess it's not that fun, but I'll say that for this particular data set, we don't yet have information on the precise localization of the electrodes because we haven't been able to get it because of COVID. So like a lot of people, haven't been able to be in the lab physically since around March. So these are the general locations, but I think this data set is interesting because there's a lot of different areas, and you'll notice that there are very different responses to these speech sounds areas. All right, so I'll pause, and I see one question from Alex. Would you mind describing a little bit about the instrumentation and software used to collect the data, i.e. number of electrodes, event recording software, etc.? Yes, so, all right, so I'm gonna mark as answering live. So I guess I said for the EEG data, we're using the Brain Vision ActiChamp system. We're using actually an old version of their software called Recorder. I know they now have some software called Recorder, I think, that is a little bit newer than the High Quarter PyQuarter is based on Python, and basically just allow record from the 64 channels. In addition to those 64 channels, we also have 2 EOG electrodes, so for vertical and horizontal eye movements. And so you'll see those in the data set as HEOG for horizontal and VEOG for vertical. And those can get rid of these sort of muscular artifacts from eye movement. And then in terms of the event recording software, like I said, for EEG, we have actually chosen to record, we send the audio through the EEG recorder, and we originally recorded a very high sampling rate. So we start with a sampling of 25 kilohertz, which is really overkill for most EEG experiments, but not for sound. So if you are trying to record the sound itself, then to get a high quality sound, you probably want at least 16 kilohertz, I would say. And the reason that we do that is it does seem to be that the time is a little bit more robust. And we're also using, instead of presenting with something like time, we actually have written custom software in our lab with Swift on an iPad. And that is non-standard, but the reason that we do that is we have been collecting data with the same iPad setup for both the EEG and the ECOG participants. So we need something kind of portable like that. And the iPad works well for that. And you can actually write scripts that will refresh according the repetition rate of the screen. So you can keep the timing very precise as well. Okay, for the ECOG data, this is a totally different setup. This is actually using a Tucker Davis Technologies system, which is an electrophysiology hardware setup that's typically used for multi-unit local field potential activity. But ECOG data, I would say anyone who works with that, it's really variable what type of hardware people use. People may use the clinical hardware. So for example, there are companies like Natus that have their Natus Quantum Amplifier and Natus Excel Tech. But one of the downsides of those clinical systems is not for, they're amazing for recording clinical good seizure related data, but for some of these, it's not possible to also synchronize an auditory stimulus or a visual stimulus in an easy way. I think that's changing. And so if others know about this, you can definitely let me know. Having those additional sort of analog ports for synchronizing stimuli is something that's typically there for research setups, and I haven't seen that as much in the clinical setting. They do, so the ECOG data, the other thing for those who are not as familiar with this, this type of recording is that this is usually recorded in patients with intractable epilepsy. And so what that means is that they have epilepsy that is not able to be treated adequately with nutrition or diet. They've tried a number of things, and so they're looking for a way to localize seizures in the brain. And then if it is so, a neurosurgeon may resect that area of the brain. Of course, they like to check first to make sure that the person would not have any potential problems with speech function or with motor function if that were to occur. So there's lots of other testing, including scalp EEG, including MEG and fMRI, that all, in my experience, I've seen have been in helping to make those sort of determinations. The way that our research fits into this is that we, while the patients are in the hospital being monitored with these electrodes, we might go in and ask them if they would be willing to into some sentences, or we're trying to make actually more fun tasks with the iPad that are a little bit more either game-like or sort of more interesting little video clips that people can listen to. And then if they feel like doing it, then we will, record with our equipment and include this stimulus synchronization through the TDT system. And if not, then, then we go. And so I think for ECOG, it's also a lot of, making sure that the participants are excited about doing the research. And if they're not, then we never want to pressure anyone to do this. So it's always just working around their other clinical care, making sure they're not in the way. And also, if we can help them pass the time and it's something that they would like to do, and that can be quite rewarding actually to work with people and spend time with them in that way. All right, that was a long answer to your question. There you go. So that's the data. If anyone has more questions about that data, feel free to add on to that. But for now, I guess we will start. Yeah, you're welcome. With looking at the data type. And I've just made this as a string. So you could change it from EEG to ECOG. I would suggest you start with EEG. And this will allow us to kind of use those data structures that I defined above to the appropriate data set or to load the appropriate event file. Okay, so the next thing, how do you get your data into MNE? So this is luckily fairly easy as long as you have your data in a certain file format. So here I am using the FIF format, which I was actually trying to look up the origin of this, and I believe these are MEG to start with. Someone can chime in if they know. Here, this is a type of file that I saved after pre-processing. However, I'll show you in just a second, they do have types of readers if you wanted to read a different type of file, like let's say a brain vision .eeg file. So let's load this. All right, it's loaded, and that was pretty fast. You'll see here that there is a flag here. This variable was just the path to our data set that I loaded from the dictionary that I made above. You can choose preload true or false. Excuse me. And that's actually going to change whether you load in the raw data or if you just load in the header. Sometimes if you have a really large data file, you may want to load it in with preload equals false. And it will load pretty much instantaneously, even if you have 5,000 seconds of data. Load really fast. You can look at things like how many channels you have, is it filtered, what's the sampling rate, what type of channels do you have? Are they EEG? Are they EOG or something else? Okay, so you can ignore, you might see this warning. MN like it if you name things slightly differently than what it wants, but it will put up with it. So, okay. So before I go on to the next, cell, I just wanted to show you that if you, for example, because I know we have a couple other folks who maybe use different types of recording systems. So you can see here, they have readers for all of these different types of software packages. So you can load read raw EEG lab. That's probably pretty typical for those of you working with EEG lab files. You can read raw brain vision. I use that one quite a lot. As I said, that's the system that we're using. But you can look through that. And so hopefully, even if you would like to try this tutorial later on your own data, as long as it's in one of those formats, then you should be good to go. Okay, so there's a lot of stuff in this structure. This is a data structure that we've named raw. And if we just print raw, then we'll see, okay, this is a raw file. Its name is MT0002 EEG.FIF. It's got 66 channels by 604,000 time points. And that corresponds to this amount of data. And this is how big it is. And this is whether the data is loaded or not. Or loaded, if you like that better. Now here, if we were to print the info structure, that has some very helpful information. So we've got things like if there are any bad channels, those would show up here. We don't have any in this particular set, but if did, they would be specified here. We also see the channel names. So for the work with EEG, these may be familiar. For those of you who are not, I will just show briefly what those kind of mean. We see how many channels that we have that are EEG or EOG, which is electrooculography. Again, those are over the eyes. A reference has been applied to this data, and we also that it has been filtered. So it's been filtered from 1 to 15 hertz. And so you'll see that in just a second, but there's all sorts of stuff here that you can check without even having to plot anything in the first place. It can also give you some helpful information. Sometimes this is in there, sometimes not. All right. So if we move on, two of them that I use relatively often are the sampling frequency and the number of channels. And those are just in raw.info sfreq and raw.info nchan. And so here I just print it. I see someone just asked a question. How do you recommend identifying bad channels data set with known bad channels to use as ground truth? So one thing I would say is that you can, there are some automated ways of detecting either bad channels or bad segments using M&E. If your channel is entirely flat, for example, and we'll see that in that, then that can let you know that that channel is probably not good. If you have a channel that looks particularly noisy, and we'll see that when we start making some plots of the power spectrum, you can see that maybe the power spectrum of all of your channels looks one way, and then you have one electrode that just looks a little bit off. If you were the person that was collecting the data, sometimes it's easier to know from the impedance measures, but with a lot of these, you may not have access to that information. So I would say you'd actually use the plots to kind of look at the power spectrum, look at the raw traces. And so we'll be doing some of that right now. So we can take a look. I think that's going to be more obvious for my ECOG data set than this one, but we'll still look through that just a bit. Okay, so let me go back here. All right, so what this is, the sampling frequency of our data is 128 hertz. And our list of 66 channels includes all of these. And it's listing all of them, including the EOG channels, which are not recording neural signals, but are actually sort of that we use to help us with artifact rejection and correction. Okay, so the next plot that I'm going to show, this is the one that was having some trouble with the newer version of matplotlib. But here what we see is I have the channels. So we've just colored the channels according to, if they're EEG, they'll be in black. If they're EOG, I'm going to color them steel blue. And then we're going to set a sort of figure output using raw.plots. I'm using show false just because if it doesn't actually matter here, but just to make sure I don't have two copies of this. And then I also use scalings equals auto, which is another optional parameter. And I'll show you, it doesn't matter for this one if you remove it. It will look a little bit different. This is now auto scaled so that everything is shrunken, or it's actually zoomed out a little bit. Compared to the other scaling. So we'll put that back. But here you can see, for example, now this is something that you would probably want to know about your data is if it's been referenced, for example. So we referenced an average of TP9 and another electrode TP10, which would actually be if you scroll down, then it's further down. So these look flat, but that's actually just because that information has been subtracted from the EEG in the referencing system. So that's not a bad channel. So what other things can you do with this? So one thing that I encourage as well when you're looking at these is you can also plot the help for anything. And this is wonderful because you can see all of the other things that you can do with it. And so one thing here is that it's only showing the first 20 channels. So we could increase the number of channels, for example, if we wanted to see more of them at a time. If this were an interactive session, then you could actually use either your mouse or the up and down arrows to scroll through this plot. So I will say that the hub in this way is not ideal for this sort of thing. This is what you would be doing if you're plotting the raw data to do artifact. But let's just try this out anyway. So n channels, I think it's n_channels. Let's try showing all of them. It's probably going to look not so good. It might be quite, let's find out. Oh, not terrible. So you see all of the channels there, but they're kind of, so the other thing that I've done here is raw.plot returns a figure handle. So if you were running this in a notebook, then you can actually set the height and width of that figure directly. If this was a pop-up, then you could just drag it with your mouse, just like any other kind of GUI. But let's make the figure height a little larger. And then take a look and see if this looks, that's a little better. All right, so now here you can see also our HEOG and our VEOG channels, which basically, if you see blinks in the data, then you'll see these sort of vertical peaks in the VEOG channel. For saccades, you'll see these sort of things that look like square functions in the data. The person's moving their eyes around. We can also do things like choose the start time. So right now this is starting at time 0, but if we wanted to set this to some other, start time. All right, then now starting at 10 seconds, for example, instead of 0. And if we wanted to do 1,000, then you can see what that looks like as well. All right. So for this, it does look like, so there may be some artifacts going on in here that are not rejected at this point, but for the purposes of this tutorial, that's actually going to be fine. The other thing that you notice here is the color of the scroll bar here corresponds to those colors that we set up before that are, which are they EEG channels or are they EOG? Okay, so that's kind of the basics of just showing the raw data. But one really nice thing is how easy it is to make a lot of plots that you would use for identifying bad channels, troubleshooting whether your data look noisy or not, and plotting evoked data, so plotting responses to specific conditions. So the first one that I will show you is the power spectral density, or E. And this one is very useful for looking at the distribution of frequencies in your signal. This I've already run, but I'll run it. And so this is going to compute the power spectral density for our EEG. You can see here, if you're used to looking at raw EEG or ECOG signals, you generally have this kind of 1/f frequency fall off. So you tend to have higher power in the lower frequencies and then a drop off as you get to higher frequencies. This particular data set, I already showed raw.info that this had been filtered, and it was filtered from 1 to 15 hertz, which we also see automatically in this plot. So it puts dashed lines where those filter bands stopped and ended. So that's really nice because you can see that, no, the raw data wouldn't actually, there's nothing above 20 hertz, but for this particular data set, this is how we filtered it. So if we were to filter it again from 1 to 10 hertz, then you would see a drop off from 10 hertz, for example. The other thing that is nice about this plot is that you can see the colors over here that correspond to where different electrodes were recorded from the scalp. And those colors actually also correspond to the plot. And if you're running this in active mode, you can actually click on each of the individual traces here and see which channel is which on that. You can also, let's look at the help for this one as well. So if you wanted, there's many options for all of these. You can choose a minimum frequency and a maximum frequency to plot. So let's say we know it's been filtered. Maybe we only want to plot between 1 and 15 hertz. So we could put those parameters in. We could also change elements of how the filtering is done. You can change the scale. So if it's a linear or a log scale, and you can also change the colors, and you can change the picks, which I will talk about in a bit, but basically the picks means which of these channels are we going to look at. So, all right, we'll leave that as is and keep going here. And luckily all of the help functions are quite good here. So, am I missing something? No, okay. All right, so the next thing I wanted to show is that for EEG, at least, there are very convenient functions for showing the location of the sensors that you're using. And so not everyone uses a 64 channel montage for EEG. Some people record from 128 channels. Some people record from 3. Some people record from 32. So, you can't necessarily assume that you have a certain number. They do have names that are mostly sensical, so that's good, and I think is somewhat different from ECOG, which it's nonsensical, can be very different from site to site in terms of, how a specific group label electrodes and what types of acronyms they prefer to use for that. Okay, so we can't really use this plot sensors map because it doesn't make sense for ECOG. And what I would say is I'm actually not going to be talking about 3D plots for ECOG here. Noah Benson's session is happening right now also. And if you want to look back at that later, he has some really nice code for plotting 3D brains that actually I worked on a project for last NeuroHackademy with a student and postdoc. And so we worked on an ECOG visualization using Noah's tool. However, there are also some ways of plotting 3D data with ECOG, but it also requires a free surface and some other additional information that I'm not including today. Okay, so if we are still using EEG, then what we can do is we can plot our sensors. And this is a very simple function. So this is just showing the montage with all 64 channels. They are named kind of according to where they are on the scalp. So C kind of standing for central, T temporal, F frontal. Then we also have some of these anterior frontal. We have P parietal, O occipital, and I think I said T temporal. And then if they have a combination kind of O, FC is sort of frontocentral. They also are generally numbered as on the left side, you have the odd channels, odd numbers. And on the right side, you have the even channels. And on the midline, they are labeled with Z. So we've got CZ is, at the very top of the head in the center. All right, awesome. So you'll see here, yeah, now we know where our answers are. That's great. And that's also very helpful for when we're trying to interpret the that we have. So thing we do is we could plot the topo maps on this, or the PSD maps on this topographic map. So maybe the power spectrum looks different, different sensors. And so we could see that from plot PSD topo. I will say in these data, it does not look like it's particularly different. So all of these power spectra sort of nominally similar. You may see that if you had a really bad channel, you would see it in this plot as well. It would just look or have that same kind of fall off as the other channels. If we don't give PSD topo any options, so if we just call it without the arguments, you'll see this is the default. The default setting is a black background with a white PSD on top. And so you can do whatever your preference is. You can also set the colors to something completely different. You know, if you, I don't know, this is going to look horrible. Okay, there we go. So you can make both really beautiful and really ugly plots depending on how you would like to do it. All right, so, all right, for this last one, I'm going to show you the PSD again, but instead of showing each electrode separately, use this average flag, average keyword, as well as area in order to take the average and show the standard deviation across the channels. And so this might be something that you would want to report if you are trying to show the power spectrum for your data in a figure without having to have every single one of your channels as a line there, because it can get unwieldy to look at. Okay. All right, so the next thing that I wanted to show you all is we can basically make a lot of these functions even more customizable by choosing the specific channels that we want to look at one at a time. And we could choose the channels by number. So they are, you know, indexed by integers 0 through 63 if you have 64 channels. However, that's super convenient because then you have to figure out, well, what number is channel CZ? And you can imagine a place where it's maybe convenient to go either way, knowing either the number or knowing the sort of code. And so here, for our choosing the channels, we use MNE's function called pick channels. So nice name. And what you have to give pick channels is you give it the list of channel names, and then you tell it which ones you would like to include in your list. So I've chosen a few of them that are kind of around the top and center of the head. And the reason for that is that we're looking at auditory responses. And for those of you who do sort of ERP work or who have read studies, a lot of the auditory evoked potentials are strongest at the top of the head, which is a little counterintuitive if you're thinking about, well, I thought that, you know, sounds were represented in the temporal lobe, so why aren't they on the sides? Well, the dipoles end up being oriented as such that you actually see the strongest responses at the top for EEG. So those are the ones that I'll be picking here. And then we will use those to plot the PSD within a certain range, start at hertz, and then going to the sampling frequency divided by 2, which is the Nyquist limit, actually. This not really be needed. We could make it lower if wanted. And also scale log. And you can already see the result of it. But here you see, now this looks a little different from before because we're using a log scale instead of a linear scale. We've also changed the number of channels, and you can see which ones those are over here. So this is, this is quite nice. So here, let me change this 1 to 15, for example. All right. And sometimes that actually, maybe this is easier for you to look at because now we don't have that additional information in the high frequencies that we've filtered out anyway. So this is a nice way to do that. Okay. All right. So this next part, this is kind of where things to get interesting. So we've plotted the raw data, we've plotted the power spectrum, and we've plotted the location of the sensors, but we have this task, which is people are listening to these sentences. Actually not much of a task because it's really just them listening passively. For some other tasks, if there are components that are a sensory component and a decision component and a reward, then all of those things could also be marked in your event file with separate ID depending on however you would like to kind of separate these. You can keep them so that they are labeled according to a trial type. So maybe instead of this EVE with, 499 numbers for the sentences, instead of that, maybe you have 3 conditions. You have an auditory condition, a visual condition, and you have a button. You could use 1, 2, 3 instead. So it's really up to you as to how you would do this. And so you make these stimulus event files, and M&E expect that these end with EVE. And as I showed you before, they contain this information about the start time, the stop time, and the event ID for each stimulus. I'm not going to talk about this, but you can also actually determine the timings automatically if you have a stimulus channel that you're recording from. So for people who use E prime and there's a trigger channel, normally be called STI 14. If you have a stimulus channel in your EEG file, you can use M&E find events to convert that into events that can be used in this. You can also find eye movements based on amplitude of the EOG signal. So you can give it the ID of the sort of channel number for EOG, and then detect based on whether there was a blink or something like that. Okay, so to read in these events, we use M&E dot, and this is going to read in our stuff, and it's going to tell us how many unique stimuli there are, and I print out a little bit of it. So you've already kind of seen this: start sample, stop sample, and event ID. There's 595 total, and we have 380 unique stimuli in the EEG dataset. Okay, so now we're going to use these to split up data into trials of a specific length. Well, we can talk about that later, but if your trials are different lengths, then you may want to figure out how to do this a little bit differently. So in this case, I'm showing you with a fixed sort of duration before and duration after the onset of the event. That's how we're going to create the epochs. But you could, in theory, make all of your epochs different lengths. You would just have to save them slightly differently. All right, I see a question from Andrew. Is there a way to be specific with epoching using response time limitations, or only epoching certain trials that are followed or preceded by other specific trial types? That's a very good question. I think I can't think of a way that is doable automatically with M&E. You certainly could do that with Python. So you could, let's see, response time limitations. You could have another file that's a list of reaction times or something like that. Like, this is how I'm imagining this context. And if the reaction time is too late for that trial, then you just remove it from your events list. So that would be a way to do that in Python. You could also loop through, because these events, it's just a list. So you can put this to an NP array, and then you can basically manipulate that NP array any way that you would like. So you could search for every time 43 comes after 105, which here they're in random order, so I bet it's just once. But not in MNE, yes in Python, although I think it would require some more programming of that specifically. All right, so, all right, first thing, I'm just gonna run this. This allows us to reject data according to the peak-to-peak amplitude. And so here, this is basically going to get rid of really noisy trials. If there's a sort of spike in the EEG data, which can happen because of movement artifact, or that's a main one. And so we just want to get rid of those. For ECOG, that data is going to be in units of z-scores. So I've chosen a threshold of anything higher than 10 would get rejected, but we'll be talking about EEG for right now. All right. So then this next part this is where we make the epochs data structure. So we have any.epochs, and this takes in a raw data structure, which is the thing that we loaded in above. It takes our list of events, it takes the time before the event and the amount of time after the event. And you can change these if you would like. And then it also gives you a baseline. So if you would like to baseline your trials, which basically means, would you like to have all pre-trial activity be normalized so that it is around 0? And in this case, you can specify just none. If you had none by itself, that's no baselining at all, no baseline correction to any of the trials. None, 0 means use all of the data up till 0. So up till the onset of the event, that's what our silent baseline is going to be. If you had some specific time point that you wanted to use, then you could also put that in directly, but the non-zero is a nice sort of shorthand for not having to specify tmin again. The other things that we're providing are just this reject, and then I'm turning on verbose so we can see some stuff. So, all right, so it found 595 matching events, and it says applying baseline correction. So if I do this again, I'll just show you. Right, so it tells you, and so you should be able to see if you're applying baseline correction, then that will show up in the epoching. And then this projection items here is not relevant for us, and also it's not setting any metadata because we haven't provided any. All right, so next we can look inside this thing, and this is another structure, and tells you this is 595 events, good and bad. So we've got good events. And I think this is due to the sampling rate. There's some rounding, right? So to get to the number of samples that would be in 128 Hertz. This is the baseline. This is how much data we have. And it says data not loaded. But that's fine. We can still do things with it. Now, the things that are here, you don't necessarily have to look through this, but I think it's interesting to do so. So here we've got the event ID, and then we've got the number of trials for that condition. So for this, we actually have a variable number of trials for some of these sentences. For some of them, we've got 2 repeats. For some of them, we've got 10. For some, we've got 1. There are ways of specifically trimming your epoch structure so that you always have the same number of trials. I'm not going to do that here, but you can definitely do that if, for example, you always want to have 5 repeats of everything and you want to throw out the conditions where you only had 3 repeats or you only had 4. So that's definitely possible to do. All right, so we'll scroll through this thing. Okay, right. So now I point out here, this is kind of a little interlude. You could just get this data out of the epoch structure if you wanted to, and you can use that with, you can do that with epochs.getData, and that will load in the data and just put it into an array that then you can do whatever you want with that NumPy array. And for the Epochs data, it will then show you, how many events and how many time points and whether it's rejecting anything based on the rejection parameters that you set above. And all right, Gwen just asked, perhaps, how can you determine what thresholds to use for rejecting epochs? That's a really good question. I don't think I mentioned it. So for that, here I've kind of chosen an arbitrarily large number. You could look at sort of the distribution of your data. So if you had this NumPy array, kind of looking at the distribution amplitudes that you have and set a based on that distribution, but this is just kind of a large number where I know that it's really huge. I can show you if you have, or what I would say because of time is, if you would like to try reject none, you could set these to none, or you could set them to either a much higher number or a much lower number and see how it affects things, and you'll be able to see the difference. But yes, great question. So I think M&E does have some suggestions for what those values might be that would work for EEG, MEG. So that's kind of what I was going off of here. All right, this next plot is really nice because we could get that data out, and the data array is 511 good events by 66 channels, including EOG, by 155 time points. And so that 3D matrix, we can do things with it, or we can take advantage of these plotting functions that Python has available. So epochs.plotImage, and this will show you, it's loading the 511 events and time points. No baseline correction applied, not setting metadata, 0 bad epochs, and it's combining the channels using the mean. If we took that out, let's see. It plots something slightly different. So it plots the global field power, which basically shows you sort of the variability across the channels. And that can be helpful to look at as well. But for most of what I do, I'm looking at kind of evoked potentials. And so the mean makes more sense here. Now, this is the mean across all 64 channels. So for our experiment, that really might not be what we want to do. This 0 onset of a sentence. And so you can see there's a little bit of this kind of N1 P2 type complex. Because it's speech, it does look quite different than if you had an auditory response to something like a click, which would look very, nicely sharp and defined. So it does, this is just the nature of sort of continuous speech data. With different types of speech sounds, is that it doesn't look, it's not going to be as tight a response. But this is nice because we can see each of the individual epochs and a heat map showing the value in microvolts of our broadband 1 to 15 hertz signal. And then we see the average of that with the standard error here on the bottom. So, and it automatically labels the time axis, it labels the epochs. It's great. We can even change things like the color map if we want to. We can change whether this appears at all. And so this is great. So, all right. So here, what we're going to do is look at some specific channels again, instead of just all of them at once. And I have a question here that I think I'll get back to, which is basically, and maybe for some of you who've worked with both methods, this may make more sense question, but for looking at all the channels at once versus looking at single channels, it's probably going to make more of it for one of these methods compared to the other. So if you can think about that, and then maybe you can test it on your own as see if that sort of matches what you see. So here we're going to pick some channels again from the top of the head, and then we're going to plot those using picks, combine them into a mean, and then use the appropriate scaling and units. And there we go. So this is now the mean across those 5 channels. So it looks a little different here. This was all of them. There's some similarities here. If we were to just choose a smaller subset of them, kind of randomly, let's see how much that changes. Okay, and I think that changed a little bit more. So you can play around with these, and I think also if you do this, I think it's going to, right, so now this is just FC by itself and the mean across trials. So these are all possible ways of looking at these data. Okay. So someone had asked in the Q&A about certain types of trials and response times. And so one thing that you may want to do is to be able to sort your trial by some particular int. And so if you had reaction times, if you had some other auditory followed by a visual stimulus, maybe that's what you would like to do. Here, I don't have that. So what I'm doing instead is we're just going to take a channel. I'm just choosing CP6 kind of at random. And then we're going to sort by the mean activity in that trial, in that channel, and then plot that image. And so you see here now, this looks a little different, right? So it's, all of the epochs, it's still CP6, but now they're ordered in such a way that you can see from 0 to 500 epochs that that amplitude is changing as you go up. So this would perhaps make more sense if this was related to a specific portion or at the trial, but still something you can try anyway. It also has this really cute little topo map shown down here. And so that allows you to see the location of that electrode. Okay, so I guess I have a few more plots here to show, but I think what I will do is, so I'll show some of these just quickly, and then I can show the ECOG very briefly and then take some questions as well. So these other plot types are if you wanted to show all of the epochs in a topographic map, again, to see if there's any difference, then you can do that. You can set the parameters for the colors. So this is the min and max value of those amplitudes and the color of the figure and the font color. So, you know, you could choose this if you wanted it in black, although then font black also doesn't make sense. So you change that around. This part, I think, is particularly interesting because this is a way of separating your epochs to compare different trial types. So like I said before, we've mostly been looking at averages across trials that are all listening to sentences. So they're sentences with lots of different talkers. And we're looking at the average evoked response to those sentences, but knowing nothing about what's actually in them. So in this cell, I've created a list of all of the event numbers that correspond to the female talker and the male talkers. And that's just using the event ID diction that we made at the very top, looking for anything where the first character of that dot wave name is F, and then appending that to a list, FEVs. And then we can basically create these sets of event numbers that correspond to the F and the M stimuli, and then epoch them separately. So here we still have our same raw, our same events, and now we have this additional kind of filter where we are specifying which exact event IDs we want to look at. So you could also just have a single one if you want to look at a single sentence, but this is going to be a list of all of the F IDs, and this is a list of all the M IDs. The rest of this is the same. It's the same T min, so time min and time max. And so I will run that. And then we can plot them just exactly the same way we did before. And we get a lot of printouts about the rejections here. But you can see that overall, these two do look kind of different. But this is not super convenient to actually be comparing the two, right? So in order to compare them, we have a very nice function called compare plot compare evokes. And for that, we take the Epochs data and we can average across all of those trials and across all of the male trials, and then use viz plot compare evokes, and then there you go. So this looks like a more traditional kind of ERP. It also shows you which specific channel this came from, and that's great. If you want error bars, which I would really push for, then you can actually do this slightly differently, where you create a dictionary where you've listed all of the epochs in a list instead of averaging across all of them to start with. And it's a little strange because you would hope that it could just take in the epochs and figure that out, but this specific only works with the evoked data type. So if we run, then we see that the two responses are actually pretty comparable. And I think with the error bar across the trials, I would say I personally probably would not interpret that between the two super strong. So what do we want to do? I think what I would like to, last couple ones are just some kind of fancy EEG plots if you are interested in them. There's also things like if you just want to plot some data on a topographic map, you can have any kind of data matrix that you like, and then you can use plot topomap and just tell it where the sensors are with raw.info and just show that immediately. But rather than doing that, what I want to do is kind of maybe take a second, see if there are questions, and then go through some of this again and show you the difference between the EEG data and the ECOG data. So any questions? I'm also monitoring the Slack and the HackMD. I think I'll keep going, but please do add your questions if you do have them. So we're switched to ECOG data type. And so I just go back up to here and change this. And then we're going to load in the data, same as before. It's quite a bit better, so it's much faster to load. And it's also, so in our EEG data before, we had this channels list, and information here. But it's got kind of different characteristics in terms of what's in the file. Okay, I did get one question. How would you compare relative values of 4 versus 128 versus 256 channel setups? When is 256 necessary? That's a really good question. I think what I've seen is that people will argue that some of the higher density setups can help you to increase the SNR of your data through averaging, as long as the noise across the channels is not correlated. Then it can help you to get a more robust signal, because hopefully when you're averaging the signals, you may be averaging, you would have similar neural evoked signals, but not correlated noise. Although if the channels are super close to each other with EEG, it's, especially with the setups that use gel, you have to be careful to make sure that your conductive gel doesn't cause a bridge between the channels. And yeah, so I think there's also the sort of cost of setup time. So depending on the population that you work with, if you are working with patients or children or older adults, you actually a lower number so that the setup is faster, and so that's too tired. But some of these setups with this as well are quite quick to set up. So your mileage may vary, I would say. And then another question from Nick. I've seen visualizations which seem to overlay EEG or ECOG on free surfer-like structures to roughly localize it in the cortex. Does that require the subject to have free surfer data and some connection to those files, or can it be done with a standard template and registration? I believe for ECOG, usually you would want a subject-specific template, a scan from that person, a T1 that you could run through Free Surfer or other software package to get that. And then people often do the electrode localization with a CT scan that's been registered to the MRI, or there are also MR compatible intracranial electrodes, so you could just immediately get those localizations with that. For the EEG, I think you can do both. I don't do this as much. I personally, and I mean, this is also my personal opinion, I believe the results more from an EEG localization if it were on the person's own brain than on a projection to a standard template. Not that you can't do it. I think you can project to a standard template, but there's so many assumptions that go into that, that if you were trying to make a claim about spatial localization that was not coarse, I'm not sure I personally would believe it. But I will say I'm not an expert in those inverse models, which is what you would do for that. Okay, what about MEG data? Is it the same as EEG, or can there be difficulties with it? I actually do not, I'm not an expert in MEG. I've never worked with MEG data, so I'm not sure that I can really answer that question, unfortunately. So if someone else would like to hack MD, then feel free to. So I'm sorry, I don't know the answer to that question. One more for HackMD. How do we interpret rejecting Epoch based on EEG warnings? Yeah, so the warnings are increased or decreased when we change the epochs male-female. It should be using the same, that's a good question. It should be rejecting based on the same peak-to-peak amplitude, and it's just telling you how many of those have been rejected. I don't believe that it's doing rejection based on averages or anything like that, so it should be similar to that. And the warning is mostly just to sort of tell you how many epochs are rejected. If it looks like it's rejecting most of your data, then either your data are maybe too noisy, or you need to reject less data.
I think that's probably a good place to stop. I need to go set up the next presentation. People might want to take a little break. So
Yeah,
everybody,
totally.
virtual applause. You can't hear at Liberty, but they're all clapping very loudly. Thanks, that was great. Appreciate it. Hopefully
Thank
you'll
you.
come back next year.
Yes, I'd love to. And if anyone has questions about the rest of this, you can go through the on your own. And I'd suggest looking at the same electrodes, see how different those are for these different sentences, because it's cool.